This video is going to continue in the muscle chapter. We had just finished labeling our micro and macroscopic anatomy of the muscle, so we're at define a cross bridge. A cross bridge forms whenever a myosin head grabs onto an actin molecule, so that we're going to be able to do the work of contraction, because that myosin head is going to pull the actin closer to the midline of a sarcomere. So if the myosin head is not attached, it's called a head. But if it is attached, we have a cross bridge that forms between the thick filament and the thin filament. Next, the anatomy of a thin filament is actually more complicated than a thick filament. There's 3 different proteins that are going to be important here. We have the blue, which is actin. The actin contains the binding site, which is this little circle that's kind of hiding behind the orange string. That orange circle is where a myosin head is going to grab onto the actin and then pull to generate tension. Because we don't want muscle to just contract whenever it feels like it, we also have these 2 regulatory proteins that are going to control when we do and do not do the pulling on the thin filament. So the analogy that I'm going to give you is we have a gatekeeper situation. Tropomyosin is the gate, and when the gate is closed, the binding sites are hidden behind it, so myosin can't grab and ratchet. So this is a relaxed muscle that is not actively contracting. If we release calcium ions bind to troponin, the gatekeeper, and that tells the gatekeeper to open the gate. And what will happen is the tropomyosin will slide off the binding site so that now myosin can form a cross bridge and go ahead and do the contraction. So the gatekeeper gate controls when myosin can or cannot grab hold of the actin so that we can generate the tension within the muscle. Here's a video that might help to explain some of that, plus where we're going next.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Hopefully that clarified how some of those components are going to interact with each other to promote muscle contraction. Next for us to define, that calcium is all important because it controls the gatekeeper, and so we have to control where the calcium is at any given moment within a muscle cell. So, smooth endoplasmic reticulum gets a name change in muscle. It is now called sarcoplasmic reticulum. This does the regular functions that smooth ER is supposed to do, but in addition to that, it stores calcium ions inside of it. When it is told to release the calcium ions by the nervous system, it releases the calcium to find troponin so that we can pull tropomyosin off the binding sites, and then myosin can grab and ratchet. Now, we need the way to be told by the nervous system what to do, and one of our issues is that this SR is really deep in a cell, and the nervous system connects way out here on the outside of a cell. So the sarcolemma of a skeletal muscle cell actually gets these little infolding tubes that go deep into the muscle cell, and next to those we get these sacs of smooth ER that sit right next to that tube. So a terminal cisterna is a sac of sarcoplasmic reticulum that sits right next to something that's going to be called the T-tubule. The T-tubule is that infolding of the sarcolemma that dives deep into the cell, because when a nerve communicates with the cell, it communicates with the outside, but we need that information to travel into the cell so that the SR will release the calcium. So that's what these do. They dive deep into the cell and talk to the SR, and when they're stimulated, it tells the SR to release the calcium. So next we're going to talk about how contraction actually occurs. It involves everything that we have been talking about. So a neuron from the somatic division of the nervous system comes down and it talks to a muscle fiber. What it does is it releases a chemical called acetylcholine. That starts a stimulatory wave on the sarcolemma of a skeletal muscle fiber, and then that impulse is going to travel down a T-tubule deep into a cell. Once we're in there, the T-tubule is going to tell the terminal cisterna of the sarcoplasmic reticulum to release their calcium. The calcium is going to diffuse out and then find troponin, which is going to change its shape, and then it's going to pull tropomyosin off the binding site so that myosin can grab actin and pull. So there's several different steps that have to happen because skeletal muscle needs to be very finely controlled, otherwise you end up with some twitches or muscles that can't contract like they're supposed to, and so we end up with all of these checks and balances to make sure we only contract when we want to. Next, the sliding filament model. If you look at all the pictures that I've shown you in the GIF that's running on the bottom here, neither the thin filament nor the thick filament gets shorter during contraction. Instead, what happens is the thick filaments pull the thin filaments closer to the middle of the sarcomere, and as a whole, as they contract, like it's starting to do right now, the thin filament slides over the thick filament. That's why it's called the sliding filament model of contraction. This is kind of just showing you in a different form everything that I have just been saying. So the thick filament is the pink fluffy looking stuff, the blue is the thin filament, and so once again you're just seeing this doesn't get shorter, this doesn't get shorter, these just slide over this. So what three things have to happen for a muscle fiber to be able to contract? One, since it's skeletal muscle, we have to be stimulated by the nervous system. So a somatic motor neuron must stimulate the muscle. Your book goes into a lot of detail about acetylcholine and how that opens up a receptor, and it talks about sodium ions, potassium ions. I'm going to leave all of that stuff in the action potential for us to discuss in the nervous system, because muscle's already difficult enough. We don't also need to learn how nerves work at this point. So suffice it to say, the motor neuron stimulates the muscle to do something. Well, that stimulation has to travel down the sarcolemma and down the T-tubules, where we're going to talk to the sarcoplasmic reticulum and tell it to release the calcium. Once the calcium's been released, it's going to find the troponin to pull tropomyosin off the binding site so that myosin can grab actin and then pull. So, in a nutshell, we stimulate, that stimulation travels down the sarcolemma. The whole point of that stimulation was so that we would release the calcium so that we could start the whole contraction process. So your book has this picture to sort of summarize what we were just talking about. So this is what's known as an axon terminal. It's the very end of an axon from a motor neuron, which we'll talk about later. It has all these things called synaptic vesicles in them, and those vesicles are just little bubbles that contain a neurotransmitter. In this case, that neurotransmitter is called acetylcholine. When an impulse travels down this axon, it's going to stimulate the synaptic vesicles to dump the acetylcholine into the synapse, which is this space right here. That's going to make the acetylcholine bind to a receptor, which is what's going to start the stimulus traveling down the sarcolemma. So what we're going to do is we're going to open a bunch of gates that are going to allow things to happen in the muscle. What excitation-contraction means is we get excited because the nervous system is telling us to do something. That has to be tied to the contraction aspect of actin grabbing onto, myosin grabbing onto actin and then pulling. So the role of those calcium ions is they're the key to everything. If the calcium does not get released, myosin can't grab actin and pull because tropomyosin is blocking the binding sites. But if the calcium does get released, it binds to troponin, which pulls the tropomyosin off so that now myosin can grab actin and then pull. ATP is also an essential component to this. If you think back to that video from just a second ago, ATP is what allows myosin to do the power stroke. Specifically, ATP binding to myosin is what allows myosin to let go of the actin and then recock for the next round of a cross-bridge cycle. So yes, it supplies the energy for the power stroke, but more importantly, it allows the myosin to let go so that we can restart and get ready for the next round of contraction. If you run out of ATP, what will happen is the myosin will grab and pull, and then it can't let go, and your muscle stays contract, and it super sucks when that happens. It's called a cramp. We'll give it another name later on too. Rigor mortis is an unfortunate event that happens after people die. What happens is, when you're dead, you are no longer actively keeping the calcium ions in the sarcoplasmic reticulum. And so what happens is the calcium starts to leak out of the SR, and what's going to happen is it's going to find troponin, it's going to bind to it. Troponin is going to pull tropomyosin off the binding sites. Well, because our binding site's open, myosin is going to go ahead and grab on and pull, and it's going to keep doing that until we run out of ATP. If your cells are dead, they are no longer making ATP, and so eventually what's going to happen is the myosin is going to grab and ratchet and then get stuck and be unable to let go. So a dead person ends up frozen in the position they were in when they died, all as a result of their calcium got released and they ran out of ATP, so the myosin grabbed and pulled and then couldn't let go. Muscle tension is what forms whenever we do a cross-bridge cycle, so myosin grabs and pulls. The load is whatever we're trying to move, plus the tissue in you that you have to move in order to move whatever you were trying to move. So for example, if you were trying to pick up an A&P textbook, that's part of the load, but you also have to pick up your hand, you have to pick up your forearm, and that all contributes to the load as well. There's two essential types of contraction that can happen within skeletal muscle. Isometric contraction happens whenever the muscle generates tension, but it doesn't actually move the load. So as you can see right here, they've hooked this up to something called a tensiometer. It measures the amount of tension that's generated whenever the muscle tries to contract. So you can see the muscle's trying to move that weight, but the weight is so heavy and it can't generate enough tension to move it. So we have contracted, but we didn't change length, we didn't move our load. So that's what an isometric contraction is. Isotonic is different. This time we do actually move the load. We can get shorter or longer. We'll talk more about that here in just a minute. But now notice our weight is smaller. We can now generate enough tension to move that. So not only did we generate tension, we got shorter as we did that. So that's an example of an isotonic contraction. A motor unit is a motor neuron axon plus all of the muscle fibers it attaches to. So essentially, your nervous system doesn't talk to the whole biceps. You have one axon that talks to these 3 cells. You have a different axon that talks to these 2 cells. That's because you don't always have to contract your whole biceps. Like if you're going to pick up a feather, you only need these 3 fibers to contract. But if you're going to go try to pick up a car off a dog, you're going to need everything to contract. So you will activate more motor units to get more cells involved in that contraction. If you hook a muscle up to a tensiometer in a lab setting, you get something that's called a muscle twitch, which there's 3 phases to it. You can see the muscle twitch off down here. Not every muscle looks the same when it comes to a muscle twitch, but there are these 3 phases that you're going to notice. So at the center of this, at time 0, we stimulate the muscle to contract. We can do that in numerous ways when it's hooked up to a tensiometer. You can drop a neurotransmitter on it. You can shock it with something, but whatever. Right here, we stimulated it. Well, after stimulation, there's a period between when we stimulated and then before we start to actually generate this tension. This period right here is called the latent period. And there's a lot happening inside the cell during the latent period. We're opening gates, we're releasing calcium, it's binding troponin, that's pulling tropomyosin off. All of that stuff happens before myosin can grab and ratchet. After that, the period of contraction is when myosin grabs actin and then pulls. And then after that, when the contraction is over, tropomyosin slides back over the binding site and the muscle starts to relax over time. So that's our period of relaxation. Now, in the real world, when you guys are doing contractions, you don't just do one muscle twitch. Like if you're going to pick up your A&P textbook, you have to do dozens of these to pick up your textbook and then keep it elevated. And so this is really something that only happens in a lab setting when you have a muscle hooked up to a tensiometer. What is a graded muscle response and how can you grade a muscle response? So this kind of goes back to, are you trying to pick up a feather or are you trying to pick up a car? You generate a different amount of force depending on the load that you're trying to pick up. And your brain actually looks at what you're trying to pick up and it does all of this math inside of it. And it says, well, I think to pick up this highlighter, I'm going to need to activate these 2 motor units so I can pick up the highlighter plus my hand plus my forearm. And so you do that and you correctly pick up a highlighter and everything is good. Sometimes your brain does do a miscalculation. And if you guys have ever picked up a box that you thought was going to be heavy, but it wasn't, maybe you like hit yourself in the chest with that box, that's because your brain looked at that box and it said, I think that box is going to weigh 100 pounds, so I'm going to activate 50 motor units, when really the box was empty, so you only needed 10 motor units. So this is something that's happening at an involuntary level based on experiences that you've had in your past. Now the way your brain determines that difference is you can change how often you stimulate a muscle to contract, that's called temporal summation, or you can get more motor units involved. That involves the strength of the stimulation and it recruits more cells to help you pick up more things. So just the temporal summation and how that's going to work. If you only send one impulse from a motor neuron, you generate this one muscle twitch and you can generate this much tension. If, however, you fire off 2 impulses, one right after another, what I want you to notice is you can generate more tension than you could with just this one response down here. Now, if you're lifting weights and you're trying to deadlift something, for example, you do what's known as unfused tetanus and you keep stimulating that muscle over and over again and you can generate more and more and more and more tension up to a certain point. Now, this fused tetanus at the end, this is the superhuman strength of, oh no, somebody ran over my dog and I'm going to pick this car off my dog. This is not something that happens on a day-to-day, and in fact, this is not good for you. It can break your bones to do it. But these other 3 are things that happen to you all day. Now, recruitment again means get more cells involved. If you work your muscles and stress your muscles, you can make your muscles bigger over time. We'll talk about how you do that later on, but that's so that you can recruit more cells and get more things involved in your contraction so you can lift those heavier objects. Threshold stimulus is the minimal stimulus that you need to elicit a response in a muscle. So if you pretend that like we hit it with 1 millivolt down here, nothing happens. We hit it with 2 millivolts, nothing happens. We hit it with 3 millivolts, well that's the minimum stimulus, the threshold stimulus, and so now we get a couple of cells to twitch within that muscle. If we keep increasing the stimulus strength, we keep getting more and more cells involved up to what's known as the maximal stimulus. If we go above the maximal stimulus, we don't generate any more tension, and that's because all the cells were already involved in the contraction. You can't make it any stronger when everybody was already working to try to make something happen. So that's what those 2 words mean. Muscle tone is, most of your muscles are partially constricted a lot of the time just to keep your joints stabilized and maintain your posture so that you're upright and things. So this isn't about active movement, this is just keeping everything where it's supposed to be. Okay, so isotonic, we talked about this just a second ago. This means your muscle contracted and it changed length. In the example I gave you earlier, the muscle got shorter as it contracted. That is what's known as a concentric contraction. So as most muscle contracts, it gets shorter and fatter as it does that. That's concentric. The flip side to that is it is possible for a muscle to contract and stretch longer. That's called an eccentric contraction, because eccentric essentially means weird. Now, what I want you to think about is a bicep curl, which is what this dude is doing off over here. And if you have a little weight at home, get a little weight. Or you can use your A&P textbook or whatever. If you start out with your arm nice and straight, and you start to bend the book up, or your weight up towards your face, well, at that point, your biceps is carrying out a concentric contraction. But as you lower the book back down, your biceps are still contracting, but now they're getting longer. So going back down is an eccentric contraction. If you have ever done any sort of physical therapy, or you have used a physical trainer to help you out with anything, they will tell you to make sure you control, like in a bicep curl, control going down too. They don't want you to just flop it down, and that's because part of going down actually uses more force. So you can generate more tension and more gains if you control it as you go back down, and that's because it's an eccentric contraction. All right, that's where I'm going to stop this one. We'll pick up with ATP in the next video.